Hello, everyone. We appreciate you taking the time to join us today. Microbial Insights is truly thrilled to have Dr. Sven LeMay. He'll be presenting for us about developing the next generation of microbially influenced corrosion detection and monitoring technologies. Most of you already know Sven. He's a brilliant scientist and an expert in this field. He's published really important information relative and relevant to MIC. Sven received his PhD in environmental microbiology from the University of Oldenburg in Germany in 2014. He was studying the anaerobic biodegradation of hydrocarbons. And Sven has a really impressive pedigree. He's worked at renowned research institutes such as the Max Planck Institute for Marine Microbiology, Newcastle University. He has over 13 years of experience in petroleum microbiology and MIC. And Sven joined ExxonMobil in 2019 as an expert for microbial corrosion. So we are thrilled to have him with us here today. Please note that this webinar is being recorded and it's going to be available in the resource section of the Microbial Insights website within the next week. Also, you're automatically going to get a link emailed to you if you registered for the webinar, so you don't have to reach out to us for that. That'll be automatic. After the presentation today, we're going to have a short question and answer session, and we'll get through as many of your questions as we can. So if you have thoughts or questions for Sven, please type them into your GoToWebinar panel in that question section, and we'll answer as many as possible. So without any further ado, I'll go ahead and turn it over to you, Sven.
Thank you, Dora, and thank you for giving me the chance to present some of the research that we're doing at ExxonMobil towards developing the next generation of MIC detection and monitoring technologies. So first of all, I'd like to thank also my co-authors here, Mohor, George, and John, who has been vital in supporting this kind of research that we're doing. And also wanted to shout out to my previous colleagues, Dennis Enning and Jaspreet Mant, who have been a major part of our research program at ExxonMobil. So just an overview about my presentation today. I will give a brief introduction into microbially influenced corrosion, or MIC, and a few slides on mitigation methods for MIC management in the field, so what we're doing in the oil and gas sector to mitigate MIC related problems. And then I will move on to actually work discussing the development of our next generation MIC detection technologies. So we'll highlight what's done currently to monitor microorganisms in oilfield operations, as well as what we're doing to identify in the area of biomarker for MIC detection, so what we have done in this area, as well as developing qPCR assays for MIC detection. And then I will move on to some field testing of this new technology. So I will present two cases here where we have successfully used this new technology to monitor biofilm microbiomes in the field, as well as planktonic microbiomes. And then I will also show a little bit the global applicability of this technology. So microbially influenced corrosion is really like a major problem for several industries in the world, but it's also major problems for our oil and gas field operations. So if you envision a pipeline environment where you have a mixture of oil, gas, dissolved acid, gas, so like CO2 or hydrogen sulfide, and then you have steel surfaces. But beside that, you have a huge amount of different types of microorganisms present in the water phase here, and sometimes also present in some of the interfaces between the water and oil. And so some of those, under the right conditions, some of those microbes can actually settle down on our steel surfaces. And while some of those biofilms that will develop over time can be quite benign and not cause any issues, some biofilms and some species of microorganisms can actually inflict severe corrosion on our steel infrastructure that can lead to millimeters of year wall loss and cause potential failures in the field. Now, MIC cost the industry or several industries billion of US dollar per year. So there's some estimates that's like about 3 trillion US dollars and global corrosion costs per year, and eventually 20% of those can be eventually linked to MIC. And yeah, we have seen severe cases in the field. And it's pretty difficult to detect MIC with current technologies out there. So the current technologies often just deals with counting microbes, but again, it's very tricky to differentiate a non-corrosive from a corrosive biofilm just by counting microorganisms. And it's really down to a limited understanding of the underlying molecular mechanism that can cause MIC in the field. So most of my time at ExxonMobil I actually spend on thinking about this question. So how can we actually distinguish corrosive biofilms from non-corrosive biofilms in our pipelines? So in recent years, or actually like pretty much since the beginning of MIC research, sulfate-reducing microorganisms have been associated with severe cases of MIC in the field, and probably more recently, methanogenic archaea have been also thought of the key players in oil field corrosion. So despite some research over decades in this area, only very recently there have been some new concepts developed and trying to understand how those microorganisms, in particular some specific species, can cause severe types of corrosion in the field. So, but beside microbial influenced corrosion, as you can see here in this comic, there are other corrosion mechanisms that we are dealing with in the field. So, as I mentioned, you have dissolved gases in those water phases here, and these can actually cause some quite high corrosion rate in the field. Especially CO2 corrosion is a big problem, but also the production of hydrogen sulfide by some of those microbes can cause issues such as sulfide stress cracking in the steel. Now, as I mentioned, there have been some advances in recent years how those microbes might cause corrosion, and especially for methanogenic archaea, it was always a mystery how those microorganisms can actually cause corrosion, because unlike sulfate-reducing bacteria, the metabolic product of these archaea are not corrosive. So methane is generally inert towards steel. So how those microbes can cause corrosion was a mystery. And then in recent years, a few studies could actually show that some specific types of methanogens can use the steel directly as their energy source. So using electrons that coming from the corrosion reaction. So you have the zero valent iron here, which due to just contact with water will release ferrous iron into the solution, and the electrons remain in the steel. And so over time, when there's no oxygen available, the corrosion reaction will slow down because the electrons will basically accumulate in the steel. So these type of methanogens that have been recently discovered, they are able to actually utilize the electrons and thereby further accelerate the corrosion reaction in this case. And this type of mechanism has been termed EMIC, or electrical MIC, by methanogens, because they are thought to kind of short circuit this kind of reaction by utilizing the electrons that are derived from the corrosion reaction here. A similar mechanism has been also identified for sulfate-reducing microorganisms. So thereby these microorganisms can couple the consumption of the electrons here from the steel surface or from the anodic reaction with the reduction of sulfate, producing hydrogen sulfide. And then the hydrogen sulfide itself can cause corrosion. So there are some other microorganisms that might not utilize the electrons directly. They can cause corrosion by just producing hydrogen sulfide as a metabolic end product. And then the hydrogen sulfide can interact with the steel and thereby cause corrosion. And this mechanism, by sulfate-reducing bacteria, it has been termed CMIC, or chemical MIC, because it's basically producing a metabolic end product that then can cause the corrosion. And then there are several other MIC mechanisms out there that might be of technical relevance, but it's less clear how they actually work in the field. So unlike sulfate-reducing bacteria and methanogens that have been frequently associated with some of the failures that we have seen. So here are some examples what microorganisms can actually cause to steel infrastructure. There are other mechanisms out there, such as the accumulation of acidic metabolites in biofilms, so another CMIC mechanism that has been thought to cause some corrosion issues. And then there are more recently thiosulfate reducing microorganism discovered in fracking operation that can produce hydrogen sulfide and then again cause corrosion by ischemic mechanism. And then sometimes lab studies also showed that biofilms can be actually protective from acid gas corrosion, so thereby slowing down the corrosion reaction. Other times there can be a galvanic effect due to your patchy biofilm formation. So you can have like a biofilm that creates like an environment that shows a little bit different redox potential than reactions that can be happening on the bare steel and thereby accelerate anodic and cathodic reaction. There can be a deposition of catalytic minerals such as iron sulfides that can actually accelerate acid gas corrosion. And then there are other mechanisms related to an emic type of system, like such as acetogenic bacteria have been linked to this type of emic, electrical mechanism. And then some microorganisms can also excrete corrosive enzymes that can accelerate corrosion of steel. So now what we're going to do when we have MIC in the field? So what kind of mitigation methods do we have at hand to manage MIC in pipelines? So first of all, a good thing is always to have a kind of clean environment, but an oil operation is generally considered very dirty, so it's very hard to keep all those pipelines clean, especially because you have a lot of different sources that can contaminate pipelines. So first of all, when we do our well completion, there are several sources that can be seen as potential contamination sources, such as drilling mud, packer fluids, frac fluids, workover fluids. And then on the production side, we have things like hydrotest water. So these are waters that are put into the pipeline just to pressure test the pipelines and see if there are any leakage before they are commissioned into service. So these water can be a source of contamination. We have seawater injection regularly done. So we are just like using seawater to repressurize reservoirs and push out residual oil. So again, source of contamination here. And then we have also produced water that can be re-injected to reduce the water footprint of oil operation just by reducing water. And then there are also open drain systems such as rainwater, wash water, slop pits, some things that can be additional source of contamination in oil field operations. But nevertheless, microorganisms are everywhere, so it's really impossible to kill all and prevent contamination. But sometimes it's very key to keep the system as clean as possible, so just like regularly having maintenance and cleaning out any residual mud that might accumulate in tanks, etc. And thereby it's believed to slow down MIC on the long run. And beside that, in pipelines, we do other internal MIC controls, such as mechanical cleaning or pigging, whereby we are launching a kind of device through the pipeline, which is basically like a huge brush that cleans out any debris and potential biofilms out of the pipeline. And we're doing a biocide batch treatment behind the pig, which is often termed a biocide pill, which kind of then kills any residual biofilms that remains and it's not been cleaned by the pig. And beside that, we're doing also biocide application in, for example, like routine operations such as a batch mode, so once a week for a few hours, or continuous injection, such as, for example, hypochlorite during seawater injection to kind of reduce the microorganisms in the pipeline fluids. And the purpose for this biocide treatment and pigging is really to reduce corrosion on the pipeline by removing any accumulation of biofilms. And we'll talk about this a little bit later. So effective MIC mitigation really requires a constant periodic biocide application and pigging. So it's not just done once and you're good to go. You have to do this periodically because you're always pumping in fresh fluids and debris from the reservoir that then might settle down in the pipeline. So you have to do really a constant biocide and pigging to keep the pipelines as clean as possible. And the reason for that is really like why we're doing both of those is that none of the treatments, so neither pigging nor biocide, is really 100% effective. So mechanical removal can be very effective, but again, there will be always residual biofilm that might be regrowing over time. So you start eventually with a corrosive biofilm, you do your killing and removal of those biofilms, so you're creating an environment that's not as corrosive anymore. But then over time, you get additional planktonic microorganisms floating in, settling down on the steel, regrowing. So you're going to build a corrosive biofilm again. So why, just a little bit more on why pigging is very important. So the mechanical removal of biofilms and biocide itself may not provide adequate MIG control. So imagine you have a mature thick biofilm in your pipeline. So if you're doing no pigging, those biofilms will grow, there will be debris accumulating and corrosion products. So if you do your biocide application, all you're doing is really potentially treating the surface of those biofilms, and you're not really reaching far beyond the first layers of the biofilms. You still remain a very immature biofilm here that can still actively cause corrosion in the pipeline. So when you do a pigging, you actually remove most of those biofilms. You're ending up with a thinner biofilm here in your pipeline. So if you're doing a biocide application, you have a much more effective treatment here because you're reducing your overall biofilm and you're just like exposing the community below all those debris, etc., to the biocide. So really pigging increases the effectiveness of MIG mitigation because the biocide can be effectively reaching down to the steel surface. And then it's also very important to eventually optimize your pig geometry. So there are different types of pigs in the field. So pig is like this kind of cleaning devices that we launch through the pipeline, and there are different types out there. It can be just a simple sponge. You can have some plastic cups that are removing the biofilms, and then you can have also something like brush pigs, which actually contains like steel brushes, which are thought to actually do a much better job in cleaning biofilms from the pipe walls. And then something even better would be something like a pencil brush that can actually reach far down into certain cracks and remove even the potential biofilms that are sitting and actually causing localized corrosion. So in the next couple of slides, I want to talk actually about what we're doing in the area of MIG detection. So as I mentioned before, it's very tricky in the field to accurately detect MIC with what is currently available on the market. So again, when we're looking at our pipeline system, what the types of samples that we're usually getting in the field can be, for example, a corrosion coupon biofilm. So some operations that we're doing, we're having actually steel coupons inserted into the pipeline fluids to get an idea how corrosive this environment is, and eventually to grow things like biofilm that we can sample over time. And then another sample that's very easily available from most operations is actually a pipeline outlet water sample. So most of the operations, you can simply go at the end of your pipeline, open a tap and collect a water sample, something very easy to do. I mentioned pig debris. So our pigging operation, so the cleaning of the pipeline actually pushes out the debris, and you can sample that and do microbial analysis on those type of samples. And from MIC standpoint, we really think corrosion coupon biofilms and pipe wall solids or pig debris are probably the most representative samples that can be collected from those pipelines to get an idea whether you have a corrosive community present. So the most common analytical techniques that we apply in the field to monitor microorganisms. A few are listed here are, for example, serial dilution, so most probable number, or what we call bug bottles. So these basically is a very old enumeration technique for microorganisms that's based on growing bacteria and then visually infer their presence. You're doing a dilution series where you're doing like 10 times dilution steps, and then growing bacteria and looking, for example, of the presence of sulfate-reducing bacteria by the formation of iron sulfides, which will precipitate out, or just by increase in optical density. As you can imagine, this technique is not very reliable. There's a high probability for false negatives. We can probably only cultivate a couple of % of the microorganisms that are present in those pipelines. And then you also have to use an appropriate medium, which might not be often formulated according to the chemistry that the microorganisms encounter in your pipeline. And then there's also the issue with contamination. So oftentimes you might actually cross-contaminate your zero dilution bottles by using, for example, the same syringe throughout the dilution series, and thereby you can create potential false positives as well. Another technique that's been used already for decades is quantitative polymerase chain reaction, or qPCR. So with this technique, we can look at counting potential genes and the type of genes that you can use. You can use either genes that are like found in all bacteria or archaea to just simply count the number of cells or genes present in your system. But you can also look for more functional genes, like for example, genes for sulfate-reducing microorganisms or genes that are only found in methanogens. Another technique that is very, very often used in the field now is the adenosine triphosphate assay, or ATP assay, which simply counts the universal energy carrier ATP and thereby inferring cell numbers in the system. This is a very, very fast technique. Portable devices are available and you get results usually in a couple of minutes in the field. But all you get is a number of microorganisms, and oftentimes this assay is applied to water samples, and then based on these numbers, a risk category for potential MIC is made. But as I mentioned before, this is not really recommended because you might just have microorganism floating through the pipeline, not even causing any or forming any biofilms. So it's there's not really any key performance indicators or any type of system that you can use to accurately detect MIC just from this type of counting microorganisms in the water sample, right? Another technique that's more used, let's say, for like failure analysis or for risk assessment in the field is microbial community analysis through DNA sequencing. So this is based on the 16S rRNA gene. And so this technique allows us to look at all the microorganisms present, all the genera that we can find in our water samples or corrosion coupon biofilms and pipe wall solids. And then it's used to maybe look for what type of sulfate-reducing microorganisms or methanogens are present and whether those have been linked to MIC. But again, it's also not very reliable because there's always a certain degree of uncertainty here. So limitations really for MIC monitoring, especially for the biofilm monitoring, which is the preferred method for us, would be that it's logistically challenging. And at some of our assets, it's technically impossible because some of those pipelines we cannot launch pig through, so there's no pigging operation done. So it's very tricky to get a representative biofilm sample from some of our assets. And then not all assets contain corrosion coupons, or they're sitting at just convenient location, but maybe not the locations that are very interesting for us, where you might have water dropping out and then corrosion happening. In addition, relying on this kind of biofilm-based monitoring for like pig debris or corrosion coupons Usually we're only getting 4 to 6 data points per year. So from a standpoint of our mitigation, if we want to optimize our mitigation methods, it's very slow, right? We're just getting 4 to 6 data points per year. So we have to wait every time a couple of months until we can collect another sample and then compare it to what we have done before. And if our changes that we made to our management in the field has been effective. Accurate MIG diagnosis in the field actually, or for any pipeline, would take several months, given that we would rely mostly on corrosion coupons and pipe solid, but then oftentimes we also do lab testing, and those biofilms are growing inherently slow. So it would require considerable resources and time to accurately make a MIG assessment and a diagnosis in the field. And despite that effort, there's always uncertainty in the data used for MIG diagnosis and monitoring currently. And I mentioned already, despite industry-wide adoption of water-based MIG monitoring, there's really no key performance indicator currently available with those type of technologies that would allow us to make an accurate assessment in the field. So how would an ideal MIG detection and monitoring technology look like for us? And this is something we have been spent quite some time in the last years at ExxonMobil to answer this question and develop newer technologies that would help us in the field. So if you imagine you have your production site, for example, like an offshore operation, you have different platforms gathering mixtures of fluids, oil, gas, water, and then all is transported through like a main pipeline to your separation and processing. This can be, for example, onshore or just an FPSO. So you have your water flowing here in this direction. And then, so if we have eventually a corrosive biofilm sitting in the pipe wall here in those pipeline, so how can we actually know if this biofilm is there? So what we think a good solution for our problem would be if we are looking for specific biomarkers that are actually formed by a corrosive biofilm, but are not formed by a benign biofilm in the field. So these biomarkers, for example, can be a protein signature or a DNA signature, a specific gene that's present in those biofilms, and then eventually through some release of certain cells, and will be released over time into the fluid stream here. And then if your water moves over this biofilm, those signatures will be collected by the water. And then eventually at the end of the pipeline, we can just simply open a tap, collect a water sample, and then look for our MIG biomarker. So with the technologies that we are developing, and then understand if we have an active MIC mechanism that causes corrosion in those pipeline, or whether MIC is actually controlled by whatever we're doing in those pipelines, so pigging, biociding. In case we have MIC detected, we can then do biocide application, pigging, to see if we can actually bring the signal down and launch us into a category that we think is green, so controlled. And by using this type of water and MIG biomarker technology, it would be really universally applicable to all of our assets, irrespective of we having corrosion coupons present or pig debris available. In addition, this type of technology, because we can more frequently sample our pipeline now using water sample, we're improving we improving upon the time that it takes to actually mitigate or optimize our management practices. So now with this type of technology, if we're looking, for example, for qPCR or some kind of in-field tests, we're probably looking at days to weeks versus months to year to optimize our management. So we're really getting a real-time optimization of our mitigation programs. We can be also more targeted with our chemical applications. So this will eventually reduce our operational costs, because we now may not treat every pipeline as frequently as we may need, or we can be more aggressive at certain locations to actually control the type of MIC mechanism better. And in addition, our research program really looks only for biomarkers that are produced if and when MIC is occurring, making this technology more accurate. So we are not just looking at any microbes that are present. We are just looking at really some of the key MIC mechanisms that cause the corrosion. And I will present in the next couple of slides some of the more molecular biological details around those mechanisms. So currently we have detected 2 biomarkers over the couple of last years. One of those is a biomarker for corrosive methanogens, which we have named MIC-H. And so, as I mentioned, methanogens are frequently associated with severe types of MIC in the lab and also in the field. So we have seen rates of several millimeters per year in both lab and field testing, and high numbers of methanogens in those biofilms. And it was really key was a recent academic study conducted with an isolate of Methanococcus maripolidis, the strain OS7. So in this scientific study, the authors could really identify a specific and unique hydrogenase enzyme present in those strain, but that was absent in similar strains that were not causing any corrosion. And the mechanism by which this hydrogenase is facilitating the corrosion is really like by acting as a mediator, and thereby so binding to the steel surface, collecting electrons, and then actually doing the reduction of protons to form hydrogen gas, which can be then consumed by the microorganisms and reduce CO2 to methane. Now, in this study, the authors also found a 12 kilobase pair genetic island, which they termed MIC island. So the hydrogenase actually set in a genetic island with other potential exporters present. So really kinds of strengthen the type of mechanism that the authors proposed here by kind of excreting this enzyme out of the cells to then facilitate the corrosion. So now in our lab test, we have found numerous times Methanococcus maripolidis. So we were really keen on understanding whether a similar mechanism could have been at play in some of the severe types of corrosion that we've seen in lab tests. So we collected biofilms from one of those tests where we actually see severe corrosion, like 1.2 millimeters per year. And if we are looking for the localized corrosion, we even get much higher rates here. We found a lot of siderite on the corrosion as corrosion product, actually supporting the hypothesis that the corrosion was caused by methanogens, because the siderite is one of the corrosion product formed by methanogenic archaea when they corrode steel. And then we also saw like quite some high cell numbers in the biofilm itself, supporting the MIC mechanism. So we collected DNA from this coupon. We did a metagenomic analysis. And what we could actually find is, to our surprise, an identical gene cluster in one of those biofilms that actually were originated from a West African oil field. So this strain we named MIC098. And you can see it's like almost a 100% identical gene cluster with a few genes showing a little bit of different sequence identities, but most of the genes are 100% identical. So we really thought that this mechanism is likely operational and has led to this type of corrosion in our tests as well. We did then a survey on like public databases to see how conserved this gene cluster is among other archaea. We could actually see that even in other genera like Methanobacteria, we could find similar MIC islands with an identity of over 90% on the amino acid level. And then together with Microbial Insights, we actually developed a qPCR assay to detect the larger subunit of this nickel iron hydrogenase, which we named then MIG-H. And we have published also the primer and probe sequences. So if anybody's interested on adopting the assay for their operations, we have actually made all this information available in one of our publications. Now, the second biomarker that we developed targets corrosive sulfate-reducing bacteria, and we named that one MIG-C. And I just give you a little bit of an overview what we believe the mechanism for those type of microorganisms is. Again, SRB since the beginning of MIC research have been one of the MIG culprits in the field. A recent study conducted from a former colleague, Dennis Enning, could actually show that some of those SRB, this Disulfovibrio phorophyllus, can cause significant corrosion that is actually of industrial relevance, and the type of mechanism this microorganisms is utilizing seems to be in a direct electron uptake from the steel, likely via outer membrane C-type cytochromes. As you can see here, so the cells, the biofilms are forming and having a specific system that they can actually use to utilize, to take up electrons and utilize them directly for the reduction of sulfate. Now, a more recent study has actually sequenced the same SRB strain IS5 and found a unique outer membrane C-type cytochrome gene cluster, which I showed here. So a lot of different types of C-type multi-heme C-type cytochromes present in those clusters, and they could also show that upon starvation, this kind of gene cluster gets expressed and could be linked eventually to a direct electron uptake mechanism. More importantly, this unique OMC gene cluster is absent in SRB that corrode primarily by the production of hydrogen sulfide. So, for example, Disulfovibrio alaskensis, Disulfovibrio vulgaris, those types of mechanisms, they use mostly the mechanism by those microorganisms is mostly due to the formation of hydrogen sulfide. So we newly developed in-house capabilities for metagenomic analysis in our lab to screen similar as we did for MIG-H, our lab biofilms and field samples that we have collected from locations with active MIC or with from corroded steel coupons from our biofilm tests in the lab. And again, we could discover similar gene clusters in those corrosive biofilms. So some of those from different locations here, West Africa, Middle East. We also did a lot of testing with Gulf of Mexico sediment in our lab, and all of those tests showed actually relatively high corrosion rates. Now, additional study that we did with our colleagues in New Jersey, they have performed some proteomic analysis on strain IS5, actually comparing cultures that were grown on steel surfaces versus the ones that are just formed on peak surfaces, and performed a proteomic data analysis. And what they could actually show is that one of those C-type cytochromes, DFE0465, was significantly upregulated in the cultures that were causing corrosion, so the ones that were grown on steel surfaces. And for that reason, we choose that one to develop a qPCR assay for this type of cluster. We also were mining our own sample archive, so we were creating quite a bit of metagenomic data over the couple of last years, and we used that as well as public databases to do the assay refinements to cover as much as possible the diversity of this gene cluster. Now, as I mentioned, we developed qPCRs assays for both of those biomarker. We have refined those qPCR assays using like over 150 MIG lab tests, and then screen those with metagenomes to get actually all the DNA sequences out and cover as much as the diversity as possible. Again, mentioned this, we did this together with Microbial Insights, and the way we're using those qPCR assays at the moment is really like a binary marker. So whenever we find the biomarker, we assume we have active corrosion happening in the system. So the way we do this is, for example, you have a non-corrosive biofilm here, corrosive biofilm here. So this is like 2.4 millimeters per year with severe localized corrosion. When we're looking at total cell numbers here, again, 16S qPCR was performed on those. We see similar biofilms, but then only in the corrosive tests we could see either a MIG-H biomarker or in this case a MIG-C biomarker. So this is really like how we differentiate corrosive biofilms from non-corrosive biofilms if we, for example, don't have a corrosion rate associated with this. So next we wanted to understand how accurate our biomarker technology actually is and if it actually improves our MIG detection. So we compared the qPCR assay using those lab tests, this 150 plus lab tests, and compared them to our current best practice, which is corrosion coupon ATP monitoring. So I just want to walk you through these type of data that you can see here. So we have here the average corrosion on a logarithmic scale, and then you have the biofilm ATP cell counts per square centimeter on the x-axis, again, logarithmic scale here. Our current key performance indicator for ATP monitoring, which is also part of an AMPP standard, is 5 times 10 to the power 5 cells per square centimeter. So this is kind of this line. And then for convenience, I shaded the area which we considered severe corrosion or industrially significant corrosion in red versus insignificant corrosion in green. And this is really based on another AMPP classification for which we have like average corrosion higher than 0.13 millimeters per year. So that's like an average corrosion over the whole coupon surface, or localized corrosion of more than 0.38 millimeters per year. So this is actually considered severe. So we used both of these classifications to judge whether active corrosion or active MIC is happening on the coupon. So now to the data. All you can see here is actually the accurately predicted ATP numbers, so the ATP numbers that accurately predict MIC, whereas you can see you have a high amount of false positives. So basically like a lot of biofilms contain relatively high numbers, but we don't see corrosion. And this is really how we use currently also this KPI. So it really is a KPI that tells you whether MIC is possible, because we see that above this threshold, we see more often MIC happening. But then you see also like in some cases, the biofilm numbers were actually relatively low, but we still saw significant corrosion that can be linked to microbial activity on those biofilms. And then these are actually the accurately or the real accurately negative biofilm numbers. So the accuracy of those ATP monitoring is about 65%. Again, high probability for false positives and false negatives in this regard. So now next we wanted to look how our biomarker samples actually performing. So in this regard, you see a little bit more broader spread of the samples actually. So here above in red and yellow are MIG-C and MIG-H biomarker numbers, and then below you see the false positives Here in gray, we have our false negatives, and then on green are the accurately predicted negative samples. So the current accuracy with this assay is actually 85%, so it could be like 20% better than the ATP assay, and there's also much lower probability for false positives and false negatives. And in fact, some of those ATP biofilms, biomarker counts and biofilms that we have seen here in white are from biocide tests. So where probably the biofilm has been inactivated by the biocide, but there was still residual DNA present that hasn't been removed. And so we sampled that, and we could detect some false positives here. And then the false negatives suggested that there are eventually other mechanisms that are still unknown and need to be discovered. And we're hoping that eventually, like these additional biomarker can increase the accuracy even further, so by maybe another 10% here for this type of biomarker assays. But 85% is already like very good for an industry perspective. But again, there's probably a more accuracy that we can achieve over the next couple of years. So these results that I showed you before just came from lab tests. So next we wanted to ask the question, can we actually detect the biomarker also in field samples? We had a few samples from the field already analyzed on biofilms, but we also wanted to get an understanding if we can be better in the field, also like especially looking for those type of biomarkers in water samples on the planktonic microbial community. So the first case is actually from an asset, from a North American asset, offshore asset, where we had some corrosion issues, and we actually collected biofilm microbiomes. So in this case, pig debris samples, and wanted to compare a pipeline that had active MIC happening at the time of sampling to a pipeline that did not have any corrosion issues as far as we could tell. So again, offshore North American field with a history of MIC. So we had 2 platforms here and 2 pipelines gathering fluids to a third one before that's going onshore for processing. Samples were collected from locations that had a suspected MIC at the time of sampling, and a location without MIC. So our suspected MIC line, we actually knew that this line here had about 52% wall loss, numerous localized features. So this is from recent inline inspections. So again, like a specialized type of kit was launched through the pipeline that can actually get you an idea of the localized features that can be detected here, and then that can be compared to previous ILI runs, and thereby you're gonna get a potential corrosion rate over time. So this is our suspected MIC line, and then we had another line that ILI did not indicate any localized feature, and corrosion was also very low. So this was our line without MIC. So we collected pig debris from both those pipelines. And first of all, we did a qPCR analysis for total archaea and total bacteria, and the total cell counts were actually very similar in both of those pig debris samples. We did community analysis as well, and just to understand what type of microorganisms we have in those pipelines. Again, we find in both of those communities potential microorganisms that can be linked to microbial influence corrosion. So for example, Acetobacterium and acetogen was found in both pipelines. Sulfur-reducing microorganisms such as Desophil vibrio were also present in both pipelines. And then also Methanococcus was found in actually very small numbers in both pipelines. So next we then applied our biomarker qPCR assays, and we could detect both MIC-H and MIC-C in the pig debris sample from the MIC positive line, but we did not detect MIC-H or MIC-C in significant amount in the pig debris from the line that did not have any corrosion at the time of sampling. So this was a very, very good result for us. We could actually show that in pig debris, it might be actually work. We can use the biomarker to detect active MIC in the field. I will show later a few more examples for this type of sampling. But for us, this was a very, very encouraging result. So we actually wanted to move on then to do planktonic testing of the biomarker. So actually using water samples to see if we can detect biomarker in those corroding pipelines. And so for this, we collected samples from one of our West African operations, so an offshore operation, again, a field with a history of MIC. There have been some publication associated with this type of MIC as well. So we collected produced water samples from multiple locations. We sampled over the course of 1 week to actually get a more kind of representative sampling done on the planktonic phase. Again, we selected locations that were suspected of active MIC happening at the time of sampling. And we also collected from 2 locations that we know there was no MIC at the time of sampling. Again, the active corrosion was determined by inline inspection, as well as operational experience and potential lab testing that we have done in the past for certain lines. So again, we have our offshore field here, different platforms, pipelines collecting and distributing fluids across the pipeline. And so we have here our pipelines that had active MIC happening. So we collected from 3 different locations, pipeline outlet samples, and then we also collected water from wellhead fluids, as well as from pipeline that did not have any corrosion at the time of sampling. So first of all, the good result, again, we did not see any of the biomarker in those pipelines that did not have active corrosion happening at the time of sampling. Neither did we find in wellhead fluids. So cell numbers in those waters were relatively low, whereas they increased in those other pipeline already. And then when we used our biomarker technology on the pipelines that had active corrosion happening, we could actually detect either one or even both biomarker in this case, and in this case, over the course of 5 days. Again, confirming that the biomarker technology can be used to monitor MIC also in water samples in the field. So next, we wanted to understand the global applicability of this biomarker technology. So these were just two examples that we had from North America as well as from West Africa. So we wanted to now better understand how widely applicable the technology actually is. And for that reason, we collected pig debris samples from our freezer archive. So we have done numerous studies around MIC in the field, MIC diagnosis, so supporting business units on a global scale. And so we collected pig debris from numerous pipelines, including like midstream operations, offshore, onshore fields from 3 different continents. And first of all, we did again like a total cell count using qPCR in this case. And then when we correlate that with what we know about these pipelines, so whether we know there's active MIC suspected in those pipelines or not, we didn't really see a good correlation with the total numbers of microorganisms present, right? So this is not a good indicator, as I pointed out now numerous times. We also did a sequencing of the 16S rRNA gene and then looked for the percentage of SRB and methanogens in those pipelines. And again, one or the other was always present in those pipelines, sometimes in higher numbers in corroded pipelines, sometimes also in relatively high numbers in no MIG line. So again, it's not a very good indicator, even using like state-of-the-art sequencing technologies to try and answer MIC questions. So next we applied the biomarker counts, and to our surprise, again, we were only able to detect significant amounts of one or the other, or even both biomarkers in those pipeline pig debris samples from locations where we know that actually MIC was suspected at the time of sampling. And again, those were both from upstream and midstream operations, and they came from 3 different continents. So we actually, the data suggests that yes, both MIC-H and MIC-C are globally applicable biomarkers, so can be widely used for different oil field operations. So just to come to my conclusions with big MIC biomarker technology. So our big MIC biomarker enable a more reliable MIC monitoring, both for pig debris and water samples, and thereby, by using water samples, we actually ensure that the technology is also widely applicable to assets that may not have the ability for pigging. So you could use water samples to circumvent the collection of pig debris or corrosion coupons. The identification of key performance indicator is still something we are working on. So currently we use the biomarker technology as a yes/no answer to allow us to make an assessment whether those types of severely corrosive microorganisms are present in pipelines. Again, this technology would allow a faster implementation for improvements to MIC management. Again, we can do a qPCR analysis now within probably less than a week versus the usual month and years that it would take to collect ATP biofilm from corrosion coupons and then do changes to our MIC management and then resample 3 months later to actually see if that has any effect on the microbial community. So this more frequent sampling really allows for real-time optimization of MIC mitigation programs and thereby eventually reducing the OPEX costs by being more effective in how we use those chemicals in the field. And then, of course, there are still additional MIC biomarkers that need to be discovered, and we hope through a collaborative effort industry-wide that we can achieve this, and then also thereby enabling very wide applicability of this technology. And with that being said, I just want to mention that we are currently advertising a joint industry project for biomarker discovery. So this biomarker JIP actually aims to, again, develop this next generation water-based MIC monitoring further. DNV is actually the managing partner in this JIP, and we have been working with Microbial Insights and DNV to kick this off. And teaming up will eventually accelerate, as I mentioned, discovery of biomarkers through using more diverse set of samples, and then eventually also ensure widespread and sustainable deployment of the technology. So actually creating a market here for the industry so that we can have access to this technology. So actually this is a little bit an older slide. I apologize for that. So we anticipated the start already happening in 2022, but actually the current start is sometime aimed for the third quarter of 2023. So in the next couple of months, we're just gathering like the last final contracts and hopefully kick off the JIP soon. If you want to learn more about that, you can contact our partners at DNV. So Susmitha or Jose can answer any of your questions. And you can also scan this QR code to actually go to the DNV homepage to learn more about the biomarker JIP. And with that, I would like to thank you for your attention and answer any questions that you might have.
Wonderful. Thank you so much, Sven. That was a great presentation, and we got a lot of comments about that. Some of the initial questions that we have, can you talk a little bit about the available standards and practices that are in place from things like, AMPP or API, and are there standards that you would recommend that people look at for the microbial tool portion?
Yeah, as I mentioned, let me just pull up these slides. I mean, there are certain API standards for like qPCR, and probably Dora, you know more better about those than I do. So what we often recommend is like the AMPP ones, because they are really dealing with the MIC type of portion here. Let me just zoom in a bit. There you go. So this AMPP TM0212, and especially the version 2018, they contain this kind of recommended best practice for ATP monitoring. The biomarker have not been included in one of those standards. We hope that in the future they're going to be part of those standards as well. But really, these two standards is what I would recommend to take a look at. So this one is more like dealing with internal corrosion mechanisms, or everything from CO2 corrosion, H2S, and maybe just a very small portion on MIC, but this one is really good to take a look at type of methods that are out there for MIC monitoring in the field.
Great. And if you guys have additional questions about that, you can always email us at info@microbe.com, and we can pull some of those standards and send them out to you. The next question that we have for you, Sven, is kind of a broad one. Has your group done any research on quorum sensing inhibitors so that you can prevent biofilm formation within the pipelines? And then also, have you looked specifically at any inhibition potential with MIC-C and MIC-H?
No, we have not looked into quorum sensing yet. The reason for that is maybe also that it's very challenging to apply that currently. So anything that we would need to put in terms of chemicals, need to put in our pipelines, would need to get through a proper qualification and needs to be registered. So this can be a very lengthy process. And I think the especially mitigation through quorum sensing is still like an ongoing activity, whereas our group is more working really closely with the business units to kind of develop this type of technology. So quorum sensing, no, we have not looked into that. I know it's an field that's actively researched, and I hope there's some interesting finding coming out in the future that we might be able to employ. But again, it might be a couple of years down the road until we get all this technology out there. And the second question, Dora?
Yeah, just specifically about MIC-C and MIC-H, if that has been something that you've looked at inhibiting those as well.
Specifically not yet, but then there is some evidence that methanogens are a little bit more tricky to mitigate. And so, yeah, that's definitely something we want to look into, so how we can actually specifically target these microorganisms. And we do routine biocide testing, and some of those biocides, they work similar good on all of those microorganisms. So there's still like active research going on and trying to identify what biocide might be more effective in controlling those. But so far, what we have seen in our lab is that really, like, I mean, the biocides are really killing everything. And it's oftentimes just a kind of dose and exposure time that needs to be optimized depending on the experimental systems that we're using. And then, of course, in the field, it's a whole different discussion. You need to make sure that you accurately monitor your biocide program, your effectiveness, that you understand your chemical dosage is actually effective in controlling the microorganisms. I mean, the lab testing is a different animal altogether, right? I mean, here we're just looking at corrosion biofilms on small coupons. we can somehow mimic and qualify one biocide might work better than the other biocide. But when it comes to the field applicability, there we really need to monitor closely in the field as well. And we hope really that the biomarker will help us to optimize this faster in the field.
Great. The next question says, it was an excellent presentation, and we know that there are a huge number of different hydrogenases that are out there. Can you talk a little bit about the difference between the hydrogenases that cause iron corrosion and those that don't? How do we differentiate those?
Yeah, it's a very good question. And I have not specifically looked too much into other types of hydrogenases and whether those really can cause the similar type of corrosion that we see with those methanogens. All I can say is that the MIG-H hydrogenase, it's a really unique hydrogenase. So it's probably out there on forming a separate cluster if you're plotting this together with other hydrogenases. I'm not sure if I have a backup. I can actually show that. No, I don't think I have. But it's really, really unique. So when you're doing a blast search against other hydrogenases, the one from this particular organism, let me just pull up this here. It's really unique. And so every organisms and every MIC cluster that we sequenced in the field, and we find those hydrogenases really almost identical to the one found in this one strain that was isolated from a storage tank in Japan. So the Methanococcus maripolidis OY7. And then the next closely relative hydrogenase to this one, I think, would be only 40% similar on the amino acid level. So there's a big gap between this hydrogenase and other hydrogenases. So I mean, I really encourage the academic research community to look closer into this and maybe help us identify and differentiate if other hydrogenases can cause similar corrosion. But if we are growing, for example, this similar Methanococcus maripolidis, we do not see this type of corrosion. In fact, we see more like what I've shown here in this example. Like here in this biofilm, we see methanogens as well, but the corrosion is really low. So the standard hydrogenases that we're finding in some of those methanogens, or maybe also some of those SRBs here, do not cause necessarily the same type of corrosion. So it's really a unique mechanism from this particular type of hydrogenase. But again, this has been only discovered, and I hope I'm gonna see more academic studies being published on this type of hydrogenase in the future.
Great. Here's a question that I think is really common is, we're looking for some of these specific markers. Do you think there's benefit in also understanding the markers that cause biofilm formation? And then second part of that question is, since there's always bacteria that are present in our system, is there a certain level that you become concerned and maybe you increase your pigging operations or your application of biocide? Do we know any of those KPIs yet?
Short answer, no, we don't know those KPIs yet. Looking for biofilm markers, it's something we are after as well, and trying to see if we can somehow correlate that and understand when biomarkers are forming. But again, these mechanisms would be similar in corrosive and non-corrosive biofilms. So maybe in this regard, we may need to look for other markers, like maybe protein markers, higher expression of certain genes that allow us to better understand the risk for biofilm formation in those pipelines. But then there are really like limiting technologies available currently to do that. I mean, if we're just looking at the qPCR results, that just shows you a gene, and this gene might be present in both biofilms. So how differentiating corrosive from non-corrosive one, or whether you have a risk for more biofilm formation, it's really tricky one, and probably we need to go more on the RNA or protein level, and then it's becoming a whole different story and how to get accurate samples in the field, which is always a challenge. preserve accurately samples. I mean, we can do that in the lab very easily, but the field, there's more challenges and active work going on, and like finding preservatives that help us with some of the logistical challenges. But yeah, KPI development currently is ongoing for the biomarker, but we don't have currently a KPI for biofilms and pipelines based on, for example, pig debris cell numbers. And again, we sometimes find high numbers and lines that do not have much corrosion features based on ILI inspection. We sometimes find very high numbers of SRB present as well, and the line still looks pristine. And in other cases, we might have lower cell numbers, so it's really tricky to identify a proper KPI just for pig debris monitoring, for example.
Okay. The next question, I know we're kind of a little bit over time, but if you can bear with us, Sven, and have time, we got 2 more questions that I think would be good to answer. So this one says, on slide 24, you showed some corrosion coupons with corrosive and non-corrosive biofilms. Did you analyze the non-corrosive biofilms as well to look at, the differences and list kind of the more non-threatening microorganisms that were present?
I believe we did, but I have not looked too closely on the data, so I don't have it up on the top of my head. All I know is that we generally find methanogens and SRB in those type of biofilms as well. So we have looked numerous times trying to identify what makes this community maybe unique, but really all the data that we have collected over the years from all these biofilms, I mean, we do routine DNA sequencing to look at the communities, and there hasn't been really like a good correlation. So we sometimes find Disophovibrios and so on. Like I have some, actually, do you still see my slides, Dora?
No, I do not see.
Oh, okay. Let me just maybe reshare those. There you go. Okay, there you go. Oh, probably like showing things and nobody sees them actually. So, so for example, this is an example of a couple of biofilms that we analyzed that have MIC, and then we looked at the community, specifically sulfate-reducing microorganisms. And so, for example, the Disophovibrio that is often used as the kind of marker for MIC in samples. Whenever you see those, you always assume MIC is happening. We actually find sometimes even higher numbers in those non-corrosive biofilms in our lab tests, also in field samples. So that really tells me that this technology, yes, it can be helpful to understand if you are somewhere in an area that might be of concerns. Do we find SRBs? But it's really not a perfect marker, right? It always tells you like, yeah, there's a lot of SRBs present, and we have methanogens present, but it doesn't really tell you anything about the corrosion happening in those systems. And so really there where we're thinking the biomarker technology makes a difference, because we're looking for this kind of specific mechanism. And of course, there's more research that needs to be done and better understanding what those biomarkers are doing, this kind of C-type cytochrome clusters. How does the nickel iron hydrogenase really work? These are all like kind of theoretical models based on like lab testing, but there haven't been like a lot of knockout studies being done, and there's some active research going on with strain IS5, I believe. So I hope there's some more clarity coming out on the mechanism. But again, just looking at the communities, we have not found a good correlation with MIC in all those lab tests.
Great. All right, this will be our last question. If you have additional questions, feel free to reach out to us at info@microbe.com or reach out to Sven directly. But either way, we'll get with Sven and get the answers for you and send them back. But the last question, Sven, is what's the aim for ExxonMobil in terms of accuracy? You mentioned an 85% 85% current rate. Do you think more markers would help to improve the monitoring program? And how do you see that adding up in terms of the effort that's needed to monitor MIC?
Yes, I mean, we always strive for like a best possible outcome, and the 85% is already good, but we believe we can do better by actually joining or joining forces and kind of tackle this industry-wide problem more collaboratively. So really, we assume that by finding additional biomarkers, our accuracy will be better. Using a more diverse set of sample might also improve the qPCR assays that we already developed, so the MIC-H and MIC-C. And again, these 85%, that's really more from what we see internally in our lab testing, and maybe we are also a little bit biased here where we are looking. So there's certainly more to discover, and that's why we have this joint industry program that we are developing over the last couple of years that hopefully will improve the accuracy for this monitoring in the field. And then, of course, we're working towards kind of globally implementing this technology and working, of course, with our service lab like Microbial Insights to have those analyzed. Yeah, I hope that could answer the question.
Yeah, it's a great answer. And thank you again, Sven, for taking time and sharing your expertise with us. It's been a pleasure, as always, to hear you speak, and I've learned a lot during the presentation. For the audience, we appreciate you hanging out with us a little bit longer. If you have additional questions for Sven, feel free to reach out to info@microbe.com. We will get with Sven and get the answers for you, or if you have any additional questions on MIC in general that you'd like for Microbial Insights to help you with, feel free to reach out to us. I hope you have a wonderful day, and thank you again, Sven, for your time.
My pleasure. Thanks for